Hello friends, today we are discussing about the topic is peptidoglycan synthesis. The biosynthesis of bacterial cell wall, peptidoglycan is a complex process that involves enzymes reaction that takes place in the cytoplasm and inner side and outer side of the cytoplasmic membrane. It is a two-stage process. First stage, formation of disaccharide peptide monomer unit. Second stage, polymerization reaction accompanied by interaction of newly made peptidoglycan material into cell wall. The assembly of the peptidoglycan unit proceeds by a series of cytoplasmic membrane reaction. The first stage takes place in cytosol. The two amino sugars that are precursors to the peptidoglycan are N-acetylmuramic acid. Both amino sugars are made from fructose 6-phosphate. First step is glutamine, which is AA amino acid denote AA amino group to a sugar. This turns fructose 6-phosphate in the presence of enzyme called glucosamine 6-phosphate. In this step, second step is the isomerization takes place. That glucosamine 6-phosphate to glucosamine 1-phosphate in the presence of phosphoglucosamine mutase. Third step is an acetyl group is transferred from acetyl-CoA to a amino group on the glucosamine 1-phosphate, creating N-acetyl glucosamine 1-phosphate in the presence of enzyme glucosamine 1-phosphate acetyl transferase. Fourth step is the N-acetyl glucosamine 1-phosphate, which is now a monophosphate, attacks UDP, which is a pyrimidine nucleotide. It has ability to act as a energy source. In this particular reaction, after the monophosphate has attacked the UDP, an inorganic pyrophosphate is given off and is replaced by monophosphate, creating UDP and acetyl glucosamine. This initial stage it is used to create the precursor for N-azine peptidoglycan. The enzyme is N-acetyl glucosamine 1-phosphate uridyl transferase. The fifth step is UDP-GLC is converted to UDP and acetyl glucosamine enol pyruvates by addition of lactyl group to glucosamine. Also in this reaction, the carbon C hydroxyl group will remove inorganic phosphate from alpha carbon of phosphoenol pyruvates. In the presence of enzyme MUR-A reaction pathway follows an addition-elimination mechanism. In the sixth step is undergo reaction catalyzed by MUR-B using one equivalent of NADPH and solvent-derived proton. This 2 electrons reduction creates lactyl either of UDP and acetylmuramic acid. Seventh step, assembly of peptide stem of peptidoglycan is ensured by a series of 4 essential enzymes known as MUR ligase. This provides for addition of L-alanine, D-glutamic acid, L-lysine, D-alanine, D-alanine in the case of Escherichia coli. Eighth step, the second amino acid residue of the peptide stem in most species, D-glutamic acid. Ninth step is third amino acid of the peptide stem is L-lysine is added. 10th step, fourth amino acid of the peptide stem is D-alanine added. Second stage takes place in cytoplasmic membrane. The UDP-MUR pentapeptide attached to lipid carrier called bactoprenol, carbon 55 isoprenoid phosphate. They are joined by pyrophosphate bond. The UDP transposes NH to the bactoprenol PP-NAM pentapeptide, generating lipid chain. If a pentaglycan interpret is required, it is creating special glycyl tRNA molecule but not ribosome. The interpret formation occurs at the cytoplasmic side of the membrane in the presence of enzyme MUR-G. Lipid chain is flipped across membrane by flippers enzyme, but this molecule transported through the membrane is still not understood. The NAM-NAS pentapeptide is attached to the growing end of nascent peptidoglycan chain that is held at the membrane by bactoprenol molecule. Then this bactoprenol donor move back across the membrane. It loses 1-phosphate, become bactoprenol phosphate. It is now ready to begin a new cycle. The picture of peptidoglycan. or the flow chart of the peptidoglycan synthesis. Thank you.